Hayati. For the
We have Dr. Somchai, and then we have the second keynote, Dr. Warapons, and then Bu Rati udah masuk belum ya? Dr. Rati is already arrived here, so
Okay.
we still have another three panelists. Dr. Vina is here. Dr. Satya. It's okay, I think we can start.
Oke, Pak.
Okay.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Selamat pagi, Bapak Ibu. Good morning, ladies and gentlemen. Distinguished Head of Research Center for Accelerator Technology, Bapak Dr. Muhammad Rifai. Distinguished Speaker, Dr. Somchai Pancharakon from SLRI Thailand, Dr. Warapon Tantanu from SLRI Thailand, and Ibu Dr. Ratih Asmananingrum, Head of Research Center for Genetic Engineering from BRIN. Distinguished panelists, Bapak Dr. R. Taufik Purna Nugraha from Research Center for Applied Zoology, Ibu Vina Amreta Laksmi STP MSc from Research Center for Applied Microbiology, and Bapak Dr. Satya Nugraha from Research Center for Genetic Engineering, and all distinguished guests and participants. Good morning and welcome to the Accelerator Talk Series 7, Mini Symposium Series 2, Accelerator Synchrotron Technology and Utilization in Life Science. My name is Idrus Abdul Kudus, acting as the MC for today's symposium. On behalf of the host, we extend our greatest appreciation to each and every one of you for being able to attend this symposium. It is such an opportunity that we are all gathering here, although to virtual. We hope to have a fruitful discussion and insight. Bapak Ibu, ladies and gentlemen, before we start, this is a agenda for rundown of the symposium for this morning. Before we start, please let me announce some notes for the participant. The first, please keep your microphone mute whenever other speakers are doing their presentation. A question session will be guided by moderator. You can use raise hand feature, and the moderator will allow you to unmute your microphone and ask directly. Or you can write your question in a chat box. And the third, a certificate will be provided only for the participant who attends. Please don't forget to fill in the attendance form that we will share in the chat box. Ladies and gentlemen, without further ado, we are now going to listen the Head of Research Center for Accelerator Technology, Bapak Dr. Muhammad Rifai, to deliver his opening remark and officially open the symposium. Please welcome Bapak Muhammad Rifai.
Okay, thank you very much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi
Waalaikumsalam.
Distinguished participants and ladies and gentlemen. So firstly, I would like to apologize for Dr. Awaludin that he has another meeting today with our chairman of BRIN, so he cannot attend this Accelerator talk. So firstly, I'm pleased that welcome to you on the mini symposium on Accelerator Synchrotron Technology and Application for Life Science. This is our second mini symposium. That the first one is for the material science, but now is for the life science. So this is the part of the Accelerator talk series number 7 right now. So firstly, I would like to thank the keynote speaker from SLRI, Dr. Somchai Tachakarom, and then and Dr. Warapon Tatauch, and also from the BRIN, Dr. Rati Asmananingrum. He's the head of the Research Center for Genetic Engineering. We hope from the keynote speaker we can have a fruitful discussion in this mini symposium. Then also we have our panelists, Dr. Taufik Purnanugraha, Vina Amrita Laksmini, STPM, Master Science, and also Dr. Satya Nugraha. I hope these panelists will have a discussion with our keynote speaker that we can get some input for our research center to develop the facility of Accelerator. As we know, our human resource quite decreasing day by day right now because so many researchers have be retired in 5 years. So we have to prepare our new generation to study about the accelerator technology, include the Synchrotron and other facilities. So we hope the application can be used in our another research center or another research organization. So this mini symposium, we also hoping that we can can develop some community user community that in the future can we have a comment, a suggestion from them to get some good excellent accelerator facility. And right now it's used for the life science because the biology of virus research and development will be used more like synchrotron technology for characterize the material of their biology sample, something like that. So I'm confident with this mini symposium will provide information regarding about the technology itself and also the application. So with this agenda, so I would like to have a very successful webinar in another 4 hours. So and also I would like to thank you for the committee that they prepare their time and then the effort to try to manage these symposiums. So once again, thank you very much for your joining to our webinar. Thank you.
Thank you, Bapak Muhammad Ropai, for the welcome remarks. Ladies and gentlemen, I hope you enjoyed the occasion so far. We are now moved to the main agenda, that is a presentation and discussion, which will be moderated by Bapak Dr. Muhammad Rifai. For that, this is the short curriculum vitae from Dr. Muhammad Rifai. Dr. Muhammad Rifai, Head of Research Center for Accelerator Technology, BRIN Indonesia, education PhD in Hiroshima University, Japan, in mechanical engineering, and expertise in material science, metallurgy, metal and alloy, superplastic deformation. And there is a recent publication in material science, metallurgy, and other. And the international activities of us is a joint research agreement with the Doshisha University, Japan, and primarily research collaboration with Kyushu University, Japan. Bapak Rifai, again, time is yours.
Thank you very much, Mas Idrus. Yeah, thank you for the introduction for our event today. So before that, we have 3 keynote speakers. That the first one is Dr. Somchai Tachakarom. So would you like to, the readers, can you change to the slide for the Dr. Somchai? So I would like to introduce. So yeah, I'm sorry, Dr. Somchai, I would like to introduce you first before your presentation. So
that's okay.
Yeah, okay, I'm sorry. Dr. Somchai is the head of the Research and Facility Department and Synchrotron Light Research Institute in Thailand. So he graduated from University of Edinburgh, United Kingdoms, in engineering and electronics. And his expertise is about application in synchrotron-based X-ray fluorescence technique on archaeology and environmental science. And his recent publication, he has 4 about phase stability and vibrational analysis of hydrogenide diamondized bilayer graphenes and at first principle investigation. And the second one is the effect of ADTA and ATA and on cadmium distribution on translocation in Penicitum Papyrus scams CF modes, and then the polymer-based microneedle patch fabricated using micro injection molding, and also the last one is microanalysis study and ancient in Wiangkalong pottery. So this is the Dr. Somchai introduction's personal data. So Dr. Somchai, you have around 45 minutes to do presentation. Thank you. The floor is yours. Oh, yeah.
Thank you very much. Let me share our screen. No. Sorry. Okay, there's some technical problem at the moment. Just a second.
It's okay. Take your time.
ต้องเห็นสกรีนผมไหมมันขึ้นอะไรนะมันขึ้นอะไรนะโอ้ใช่โอ้อะไรนะเขาให้แชร์แล้วแต่ว่าทีนี้ปัญหาคือผมหาไอ้ตัวที่จะแชร์ไม่ได้คือผมเปิดAnd let me change my computer at the moment. I come back in a few seconds.
It's okay.
ทำไมต้องมีพาสเวิร์ดผมใช้ให้น้องเข้าอีเมลน้องเนี่ยในนี้ดูไหมเนาะกดวางไปเลยต้อมใช่ไหม
Dr. Somchai, if you don't mind, you can send your file to us, then we will try to share the screen in here. So I think Dr. Somchai will try to use another computer to present his slide, I think. So please wait a minute.
Dr. Somchai, he got a technical problem with
Oh.
his iPad. So
Ah, okay.
he very sorry about that, and he disconnected, and he will change the computer.
Okay, it's okay. We can wait. Just don't worry. Or Dr. Warapon, I'm sorry. Is it possible to change your time to Dr. Somchai? Maybe Dr. Somchai will be the second key, speaker. Ah, okay.
Okay, so sorry for the technical problem. I'm
It's okay.
using
That's okay.
Dr. Walapon computer at the moment.
Okay, it's okay.
Okay, let me copy the file. Okay, can you see my presentation?
Yeah, we can see your slide.
Okay,
Okay, thank you.
I can't see my. I take it off first. It's still running. Okay, let me share this one. มันเป็น full screen ยังไง Okay, here we go. Can you see my presentation now?
Yeah, we can see your PDF slide.
Okay.
But can you enlarge it, the slide? So maybe we can have a full screen view, something like that.
It actually already full screen.
Okay. Then because we just have with the like some web browser, and then we have a PDF files. Is it correct? Before we have the PowerPoint slide, but now we have a PDF.
Yes, now it's PDF.
Okay.
Yes, because I have some problem with the PowerPoint presentation.
Okay. Then the PDF file. So,
Okay, yep.
Okay.
Okay, let me start. Okay, good morning, everyone. I would like to, today I'm going to talk about our facility, the Synchrotron Light Research Institute, current situation, and also our future plan for the new machine. Okay, this is the topic of my talk. I'm going to talk about our institute, the Synchrotron Light Research Institute, and also our mission, our activity. Then I'm going to introduce the synchrotron radiation and its properties, the beam line, our supporting facilities apart from the beam lines, and also activities that we have been doing so far, and also our project, the SPS2. Okay, about SLI, okay. SLI, or Synchrotron Light Research Institute, is a public organization under the supervision of the Ministry of Higher Education, Science, Research, and Innovation. This ministry is quite new. It's actually only 2 years old. Before, our institute is belong to the Ministry of Science and Technology, and then we have been merged with the Office of Higher Education, become the new ministry. And we have the machine, so-called SAM photon source, which is the synchrotron radiation source. And our SAM photon source is located in Nakhon Ratchasima province, about 250 kilometers from Bangkok, or about 3 hours driving. And actually, we have an airport in Nakhon Ratchasima, or the nickname is Korat. But unfortunately, the airport is like on and off situation. Now it's already open just this year, but they have only a few flights a day, so it's not very convenient for our user. But I think there's going to be some development pretty soon, I guess. Okay, this is the bird eye view of our facility. Our you can see the round shape building, which is the building that contain our facility, our source, the SAM photon source. You will notice that most of the synchrotron facility have the similar shape building, because mostly synchrotron source are circular accelerator. I say that because there's one generation, the fourth generation of synchrotron source, it actually the straight line we call free electron laser. That's you will see some building that's actually a straight line. It's also the synchrotron radiation source, but mostly circular shape. Oh, we are in the SUT campus, Solanali University of Technology campus. They loan us our some land to build our facility. And this is some detail. Okay. The Synchrotron source.
Excuse me, Dr. Somchai,
Yes.
the slide is doesn't move the first slide, the cover slide.
Oh,
You
it's-
try to change it?
It's not moving.
Not moving actually. I thought you still talk about the first cover. Okay, this is already changed.
Okay. Okay. Have you seen this one?
Yes, we have.
Okay.
Yes, just now we just saw it.
And this one?
Yes, just now we
Okay.
said. Okay.
This one is like I mentioned, is the you can see the yellow circle. That's the main building, the one that we have the source, the Synchrotron machine inside the building, and most of the Synchrotron facility have the round shaped building. Okay, the Synchrotron source is the first Synchrotron light source in Southeast Asia, and also in Thailand, and it was the machine was donated from Sotec Corporation in Japan. They use it as a machine for lithography technique. It's a I think it's a electronic companies, group of companies, and they used to build some I think integrated circuit, something like that, and they use it for quite some time, and then they donated the machine to Thailand, and then we modify the machine, quite major modification. We expand the solid link to have more beam lines, and also to get higher energy, so we can expand the spectrum of synchrotron radiation, go to higher energy, and also increase the electron current, so we can have more intensity of synchrotron radiation. And SRI is a user facility. That means we open for public users to come and use our facility for free. But there's a competition, because the beam time is quite limited. So if the proposal to use the beam time is more than that, we can serve. They might have we have to select according to the scientific merit. And we started to do the service in 2003 with the photo emission spectroscopy beam line. And after that, we continue building the new beam lines like every year. Today we have 11 beam lines available for service. And usually before the COVID-19 pandemic, we have about 400 projects a year from local and international, both from academia and industry, to come and use our facility. And we also organize some activities like science camps, workshops, and training, about more than 20 activities per year. This is the model of our facility. It's like I think Dr. Wutikai might mention already in the talk last week, but I'm going to briefly mention the main components of our facility, our machine is that first we have the electron gun to produce high intensity of electron beam, and then we chop the electron into bunches, and then accelerate the electron bunch with linear accelerator from low energy to about 40 MeV, and then we transport the all electron bunch into the booster synchrotron booster ring. These two components install underground to protect the radiation, unwanted radiation, and then the booster ring will accelerate electron to 1.2 GeV, and then we inject electron bunch of 1.2 GeV up to the the ground floor into the solid ring, where we keep our electron as long as possible for users. And the solid ring is installed behind the wall, behind the 50 centimeters thick concrete wall to protect the radiation. And we run the machine in we call it in injection mode or decay mode, which means that sorry. Sorry, I just recovered from COVID like 2 weeks ago. Still some symptom left. Okay. Our machine is running in decay mode, which means that we inject electron beam into solid ring twice a day at 8 o'clock and 8 o'clock in the morning and 8 o'clock at night, and to the maximum current that we can. So far, we can inject the electron beam to the solid ring to 150 milliamp and let it decay until 8 p.m. and then we re-inject. We add up electron to 150 again. So we run the machine 24 hours a day. And in normal this situation before the pandemic, we run the machine about 9 months a year. And we service for user about 4,500 hours a year. But after the pandemic, like for example, this year, we still have some problem with the regulation. The user cannot come to perform experiment. We have to do the mail-in scheme, and even domestic user cannot come to our facility. That means we have to do the experiment for them. So we have to limit the beam time. Okay, our synchrotron facility is actually one of the about 50 synchrotron facility. This picture is like some example of the synchrotron facility around the world. I should highlight that our facility, our synchrotron facility is 1.2 GeV of electron energy. It's quite small comparing to other facility in like the Western country or in developed country like Japan. Japan, they have several facility. Like you can see, the right hand side is 8 facility only in Japan, and the biggest synchrotron facility is also in Japan. I think it's in, I don't remember that. they call spin 8. The electron is 8 GeV, which is the biggest synchrotron in the world. And in Southeast Asia, there are another one in Singapore, but it's belong to the National University of Singapore, which means that they're mostly doing research within the university or collaborate with other researchers. This is the mission of our facility, our institute. Firstly, we conduct research using synchrotron light and related techniques. Conduct research means we do some research ourselves, but it has to align with the national interests, as well as collaborate with users. The second mission is we provide synchrotron light for service. As I mentioned, we are user facility. User from academia or industry can submit a proposal and come to use synchrotron light, and then go back to their institute to analyze data, something like that. It's free of charge. And also our third mission is to promote the usage of synchrotron radiation, because our budget is come from the government, come from the tax. That means we have to provide our facility as an infrastructure for the country. And also we do the technology transfer to industry and to public whenever needed. This is our the main area that of our research so far. Mostly we focus on food and agriculture because it's a national interest at the moment. And as I mentioned, we do the service for users. We open the facility for users. All the users from public sector or industrial sector, both in domestic user and international. There are two types of research that can come to use our facility. The first we call non-proprietary research. This is this type free of charge. Anyone can submit the proposal to use the beam time, but of course, all proposals have to be reviewed from the committee, and then we allocate the beam time to user, and then when they get the beam time, they carry the sample, come to use our facility. This is free of charge. Another type of research we call proprietary research. That means all the data is secret, is confidential. Like mostly this type is come from the industry, private sector. For this research, we usually reserve some beam time for this type of research, because we know that their project or their research is quite urgent, because it's a competition in the market, so they cannot wait for very long time. And of course, we will charge some beam time. This is the statistic of our. The left one is the number of projects. As I mentioned, that we have more than 400 proposals per year before the pandemic. And the right one is a number of users since 2017, because it's the year that we started to receive a proposal or user from abroad, from ASEAN and also from in other countries. Okay, synchrotron radiation and its property. I think you may know that the synchrotron radiation is actually the electromagnetic radiation from the high energy charged particle, such as electrons, when it's bent or change the direction in magnetic field. Mostly in particle accelerators. Not necessarily that has to be electron. It can be proton, but the problem is proton is 2,000 times heavier than electron. It's difficult to accelerate. Like in CERN, they use the proton and antiproton accelerate to really high energy because they have another purpose. They also have a proton synchrotron as well, but the main purpose is to to study the particle. It's different purpose. Okay, so mostly most of synchrotron facility is electron synchrotron. Okay, this is the general components of synchrotron source. I think I'm going to skip this one. Okay, we started from the electron source and push the synchrotron to start accelerate to high energy, and then store the electron in the solid link to keep it as long as possible. And when electron is bent in magnetic field, it emit radiation, and where we can install so-called a beam line to bring synchrotron radiation from the bending magnet or insertion devices to the end of the beam line, where the user can put the sample and perform some experiment. Okay, this is the specification of our synchrotron. Electron is 1.2 GeV and circumference is 81.3 meters. The source of the synchrotron can come from the bending magnet, the blue one, which is the dipole magnet. You can see the spectrum of the synchrotron radiation from bending magnet and from different source. The synchrotron radiation, the energy and the intensity depends on the electron energy and the magnetic field. The higher electron energy, the higher intensity of synchrotron radiation, basically. So if you need higher energy, we need, because we the machine, we cannot increase the energy of electron. That means if we need higher energy or high intensity, we need to put more magnetic field. So you can see the spectrum. If we install the wavelength shifter, which is the dipole magnet with 6.5 Tesla, so we can increase, we can shift the spectrum to higher energy as well as the higher flux. We already install so-called superconducting multiple Wiggler with magnetic field of 3.5 Tesla. It's a multiple Wiggler. That means it consists of a series of dipole magnet. Electron can bend several times, can we call Wiggler. is so it's bend, and then is the synchrotron emitted from each bend of electron. So electron is in the synchrotron radiation is intensity is increased to very high intensity. So far we have 4 insertion devices in our solid ring. So as I mentioned, the synchrotron radiation source is mostly a circular particle accelerator, similar to cyclotron, but different principle. The magnetic field of the synchrotron machine has to increase with the increasing of electron beam. As I mentioned in the booster synchrotron, electron, in our case, electron is they are accelerated from 40 MeV to 1.2 GeV, and we have to keep electron in the same path, same radius of same trajectory. That means we have to increase the magnetic field of bending to keep electron in the same direction, in the same trajectory. That we call it is synchronization between the increase of electron energy and the magnetic field. And the synchrotron beam, it actually is broad spectrum. It's covered from infrared to X-ray. If you have higher energy of electron solid ring, you can go even higher of hard X-ray. Like in our machine, the maximum energy of X-ray we can get is about 30 keV, and the lowest is from the infrared beam line. And if you go to spring it, they can perform very hard X-ray experiment, like 80 keV or 100 keV experiment. And also the as I mentioned, if the flux of the synchrotron radiation is very high, and also the divergence is very small, that means in some techniques or some beam line, we need very small beam. For normal source like X-ray tube, the divergence is very high. That means when we focus the X-ray, we cannot go to very small beam because it will lose most of the X-ray photon. In synchrotron case, because the angle is the divergence is very small, we can go to for example in beam line 5.1 of our beam line, we can go to 30 micron in diameters with usable intensity. And even if you go to, for example, in Taiwan, they have the 3GV machine. One beam line, they can go to even nano scale, like 50 nanometers beam size. They can do the nano probe experiment. This is the comparison between source of general source and also the synchrotron radiation source. You can see the development of generation of synchrotron comparing to X-ray source from X-ray tube. Our machine is second generation. Actually, it's a modified one. It's between second generation and third generation. The difference between second generation and third generation is the emittance of electron beam. So for second generation is only bending magnet with no insertion devices. We cannot go to very small emittance. That means we cannot go to microbeam. So in our case, we modify the solid link that and enable us to install some insertion devices, so we can reduce the emittance of electron beam to, I think in our case it's about 60 nanometer radian. So we can go to micron scale of electron beam. And fourth generation, there are actually two types of fourth generation. One is they call free electron laser. It's a linear accelerator with very long line of accelerator with series of undulator, and they use free electron to generate laser in X-ray region. Another type of fourth generation is a circular one. They call it diffraction limit synchrotron source. It's still circular accelerator, but it's beyond the limit of third generation. The emittance is very small. It's like 1,000 times lower than the third generation. That means the intensity of synchrotron can go up to 1,000 times. Other properties is high stability, tunable, because the as I mentioned, it's a broad spectrum of electromagnetic radiation. We can select the energy of synchrotron radiation, depends on the beamline, depends on the techniques that we use, that we require. For example, if you would like to study the crystal structure, you need X-ray. If you need to study like surface, you might need low energy X-ray or soft X-ray, or even vacuum and ultraviolet. Or if you need to study molecular structure, you can use infrared, for example. And the synchrotron radiation is actually polarized light, because it comes from the magnetic dipole. Is fixed the direction of the magnetic magnetic field. So it's actually the synchrotron radiation is linear polarization. It's linear polarized light, and in some cases, like in some facility, they install the undulator, the hybrid undulator. They can modify the polarization of synchrotron light to be circular polarization, circular polarized light. Another property is the pulse is very short because of the electron bunch. As I mentioned, we accept in the solid link. It's actually not the continuous beam. It's actually the electron bunch. The bunch is very small in femtosecond. So in some case, they study the timing. They need a very short pulse of X-ray beam. the beam lines at our facility. So far, we have 11 beam lines available. Actually, 2 beam lines is under commissioning. The beam line 5.1 and 7.2 commissioning is nearly finished. Okay, there are 2 categories of beam lines. The first one, you can see the first box, the orange one. It actually is the insertion beam lines. We use the synchrotron radiation from its insertion devices. And another category is the bending magnet beam lines. They use the synchrotron radiation from bending magnet. And I like to mention some of this beam line. You can see some logo like beam line 5.2 X-ray absorption. It's a contract beam line. It's a collaborative beam line between Solanelli University of Technology, the Nanotech Center, and SRI. So we all invest some budget to build this beam line because it is very popular and high demand for this technique, for this X-ray absorption. So we've been collaborate with these 2 organizations for I think more than already 10 years. Another one is beamline 1.1 W, the multiple X-ray techniques. This is another model of collaboration. It's from Concan University. They signed a contract to reserve 50% of beam time from this beam line to use the beam 1.1 beam line, and the requirement of Concan University is different because they have many researchers from different fields, so they need several techniques. That's why beamline 1.1 has X-ray fluorescence techniques, X-ray absorption techniques, X-ray diffraction, and as well as glazing incident, X-ray diffraction. And they are commissioning X-ray, I think reflectometry to study the multi-layer coating, something like that. And also you can see beamline 1.3 W, small angle X-ray scattering. Actually, one company, SCG, Sam Cement Group, they collaborate with us to use the beamline. The contract is they give us the very sophisticated detector, about 20 million baht to use in this beamline, and the detector can be used by public user, and they reserve about 6 days a month for 5 years for this particular beamline to conduct the research on polymers. It's one of the company in this SCG. It's called SCG Polymer. Okay. I like to mention one by one beam lines, but I'm not going to go into detail because it quite takes some time. The beam line 1, there are 3 of them, like 1.1, 1.2, 1.3. They actually come from the same insertion devices, and we divided the beam into 3 beam lines. 1.1, as I mentioned, there are several techniques available for study the mainly for materials to study the for example, the chemical oxidation state of some elements, to study the elemental composition of the samples, and they have also diffraction to study the crystal structure, and it's a powder diffraction. The second beamline is X-ray tomography. This beamline can use to study the 3D structure of the sample. The beam size is very small. It's similar to CT scan in the hospital, but the beam size is very small. It's suitable for small sample. Okay, Dr. Varapon will explain a bit about some application using this beamline. The beamline 1.3 is X-ray scattering, or small angle X-ray scattering, and wide angle X-ray scattering. This one is similar to X-ray diffraction, but it's for larger structure, like in nanometer scale. For example, in polymer, like I mentioned before, for SCT chemical, and for study structure of fiber, or something like that. And the 2.2 beamline 2.2 is time resolved X-ray absorption. This is another absorption beamline, but in this one, the detector can collect the data in very short time, so we can track the sample in very short time. For example, in battery research, you can do the
I'm sorry,
charge.
Dr. Somchai, another
Yes.
10 minutes left. 10 minutes left.
Okay. Okay. I will do quicker. Okay. Okay. We also have the beamline for study the surface of electronic structure, the PS and PIM. We have the IR infrared to study the molecular structure and X-ray absorption, as mentioned before. We have micro XAS. Sometimes we need to focus on particular area of the samples in some in homogeneous samples, or do the distribution of X-ray fluorescence to study the distribution of element on the sample. We use the micro XIF. We have the beamline 6 lithography to build a micro structure like gear, or very small structure like microneedle, as you mentioned in one of my publications. We have a protein crystallography beamline for protein to study the protein structure. Supporting facility, apart from the Synchrotron beamline, we have a supporting facility like we have general equipment like SEM, XRD, XIF, and we also have the food agriculture laboratory to focus on this particular research. We have sample preparation lab for user to prepare the sample before going to the beamline. We have protein crystallization lab with the robot to help protein researcher to crystallize protein. And as Dr. Avutika mentioned last week, we have computational lab for computational science research. We have machine shop and technical support and engineering support. To sometimes we need to build our own equipment or chambers. we have cryogenic plant because we have some insertion devices to be need liquid helium. We have optic lab to this one is long profilometer because the we need to measure the curvature of very long mirror like X-ray is 1.2 meters long. As I mentioned, we have user facility that means we have to organize activities for users. So basically, we have before the pandemic, we have ASEAN Synchrotron Science Camp that we organize every year until 2018. That was the last one. I think we are going to organize the next one probably next year. And we also organize a series of workshops, ASEAN workshops and international conference. Like this year, the country has already opened, so we organize an IPAC, the International Particle Accelerator Conference in Bangkok, and also the Asian Conference on X-ray Absorption in Pattaya last month. Okay. We also have some internship program. There's some good example that we have a student from Indonesia to stay with us for some time to do their research. And we also have several activities like training, user meeting. Some of them are in Thai and some of them are in English. Okay. The last topic, the next generation synchrotron radiation source is a so-called photon source, the second one. Okay, so it's fourth generation synchrotron radiation. We are going to the project is already approved by the government, but we still have some problem with the budget so far. But it's still in the a little bit late from the timeline, but still on the right track. It's going to not in Nakhon Ratchasima, it will be in Ilayong province, but it's the better location because it's close to the industrial estate and also close to the beach, so the user can enjoy the beach after the long experiment. And this is some parameters of the new machine. It's the energy is 3 GeV. Okay, this is okay, and the structure of the magnet is I think it's a we call double triple bend acomet. The one sector consists of 3 bending magnet with double, as you can see the picture below. We are going to have 7 beam lines for the first phase, and probably the next beam line after the first phase will be like 1 beam line a year. And this is the comparison between our current machine and the next one, from 1.2 to 3 GeV, and the beam current will be from 150 to 300 milliamp. So the intensity will be really high, and the operation mode will be top-up mode. So we don't have to inject every 12 hours. We'll be top up every 1 minute or a few seconds. So the intensity of synchrotron radiation will be constant. Okay, and we can have more beam lines, like 28 beam lines in total. This is the intensity of synchrotron radiation. The right one is from the SPS2. It will be really, really high. Okay, And for this project, our strategy is to try as much as possible to machine all the components locally with using our local company. And so we have to do collaborate with the local company. We design our magnet by ourselves and construct the magnet in-house and fabricate the chamber by ourselves in our machine shop. This is the building of our new facility. It's going to be really modern. Okay, this is the timeline. So it will be the plan is we finish in, I think it's about 2027. So when we have the first beam line. Oh no, I think it's about 2029. The first light. Okay, this is all my talk. Thank you very much.
Thank you very much, Dr. Somchai, for your nice presentation. It's quite interesting, actually. Dr. Somchai talked about the structure of the SLI, the synchrotron, the walls, and also the management of the facility itself, and also have a new project in SPS too. Quite interesting. He has another project from the 1.2 giga ethervolt to 3 giga ethervolt. It's quite chance for us, maybe from the life science department can use this kind of facility to characterize their material. Okay, we can move to the second speaker, Dr. Waraporn. So before that, I would like to introduce her. Mas Idrus, can you share the personal data of Dr. Waraporn? Okay. Our second keynote speaker is Dr. Waraporn Tatanuts. She's worked at the Synchrotron Light Source Institute as a beamline scientist, and her expertise will be FTER microspectroscopy. And she has for recent publication regarding the performance evaluation of focal plane array, FPA, and FTER, and also synchrotron radiation with FTER microspectroscopy to classify rice components. And then the second one is artifact from Bang Chiang Talent about the pottery with hematite red geometric pattern. And the third one is about the anti-cancer activity in Laddernia crustacea, LF male-fars crustacean on human HCT116 colon cancer cells via cellular lipid and beta acid protein accumulation. And the last one is about FTER microspectroscopy for the assessment in microplasma in Hep G2 cell culture. So Dr. Waraporn, you have 45 minutes for the presentation, and the floor is yours.
Okay, thank you very much for your introduction. I will share my presentation.
I'm sorry, we cannot see your larger slides. Yes. Okay, we can see the larger slides.
Okay. Good morning, everyone. I'm Waraporn Tantanut. I'm a beamline scientist from SLRI. Actually, I'm at Asian beamline, and today I will tell you about life science application. So I don't know how about your background, but mostly I would talk about like food application and some of medical science and some in environment. So if participants have more questions about the other related topic, so you can ask me after presentation. You may know that a synchrotron, there are a lot of technique to measure. So this is like the advanced technology. You can use, you can measure sample by not need to do the extraction. So it's called non-destructive analysis. So if you have sample like rice grain, you can bring a rice grain and measure it directly, and you get a lot of information. What kind of the information that you can get? You can get chemical composition. You can do the characterization with the microstructure. So you can see what inside beneath your surface, and you can get the information of nanometer structure, like crystal structure of, suppose in this one is a rice flour, you can get the crystal structure as well. And you can go deep down into the Armstrong level, like if the sample contain any interesting element, you can measure what kind of element inside and what is the element, the neighboring element, and the bond length of neighboring element as well. As Dr. Simchai mentioned you, as present, we have 11 beamline, and there are 7 beamline is suitable for life-size application. This is the X-ray absorption spectroscopy, small angle X-ray scattering. Have you seen my mouse pointing at the slide? Anyone see my pointing?
No.
No. Okay. So other beamline is like infrared microspectroscopy. So I will start with the first beamline is beamline 1.2 X-ray imaging tomography microscopy. This beamline, you can get the 3D structure of your sample. So we will shine the X-ray beam, here this X-ray beam, into the sample. Is it passed through the sample to the detector? So we will get the image from X-ray. We will get the X-ray image in one direction, and we have the rotational stage at 360 degrees. So we will change the position of the sample 1 degree by 1 degree, and we will get a lot of X-ray image. Then we will combine this image all together to reconstruct the image of 3D structure. And we will get the information of sample by not need to do any section. Anyway, sample have to be very small because the beam size is about 3 millimeter. So the sample have to be small enough to put in the rotational stage. For application of this beamline, for example, like for this one, you can do the product development, like do the extruder for snack using a flour, different kind of flour. When we mix with different concentration of wheat barn, you can see the texture of the sample is totally different. So we would like to see the void air inside. We can measure the void air volume as well. And for shelf life analysis, if for this one is the corn meal extruded, stored at 10 days in different moisture condition. So if the very low moisture condition, the void volume inside or the air volume is increased significantly. So you can use this one to analyze. How to analyze the volume? I give you this example of a geopolymer. This one, when we do a 3D structure, after finish, we have the program to do the analysis further. Like for this one, the pore size, the pore have different shape, like a closed porosity, open porosity. So it's different concentration. We can see the percentage difference between sample. And also, this one is a sample of rat bone. We can do the 3D structure of it, and we can do further section inside of the rat bone as well. And this is the cellular X-ray image. This is kind of 2D, but we can show the 3D of the cellular as well. So this one is very useful to see deep down into the microstructure of sample in any kind of research field. And if you're interested in and you don't know that your sample is suitable to use this beamline or not, you can ask me later. And this one is the small angle X-ray scattering. It's called beamline 1.3. This one gives the information of sex and wax. And this, you can choose which technique that you would like to get the information. For this one, small angle X-ray scattering, it can be used to determine the shape of the sample, to see the size, how big of it, the size distribution, how equal of the size in it, how many size in your sample. Like for my cell, if there are different kind of my cell in the sample, it can be distinguished by the spectrum. It will show the difference between different size or shape of sample. And also the wax wide angle X-ray scattering give the other information. It can give you the information of crystal structure, not only the size. So for example, this is the chocolate. So they would like to see the proper temperature to do the nucleation of chocolate. So if we want to get the smooth chocolate, the shape of nucleation should be like a type V. So they try to use different kind of temperature to see the pattern of wax pattern. So this one is very useful for any sample that have crystal structure, like rice flour or any kind of flour, potato flour. It have the crystal structure or the crystal in the particle of the flour as well, and it's packing in different shape. Like for rice flour, the crystal packing can be like an A shape or B shape. For the A shape, is packed very tightly. So the spectrum, we will get the spectrum of crystal structure very different from the type B. This is the loosely packed and have the hole inside. And if the sample contain both of lipid and a chain of amylose, then it will be V type of the mixing. Okay, for the next beam light is X-ray fluorescent, XRF, and X-ray absorption spectroscopy. You may be familiar with the analysis of element, and this both technique can be measure element by not need to do the extraction, not need to use any chemical to see what kind of element in your sample. And it gives the information of the oxidation states of sample as well. So in SLRI, we have 4 beam light that you can apply to use. Every beam light can analyze this element from silicon into AC. Almost element in this table can be measured unless only the low element at the top cannot be measured. So for this beam light, any kind of sample can be measured. Your sample, if your sample is the liquid form, it also can be measured directly. And if sample is the tissue, the plant tissue, you can choose beam light 5.1 Asian beam light to measure. Or if your sample is the grain, the powder, the particle, it can be packed in the pellet and measured directly as well. So that, as I mentioned to you before, that the conventional analysis of element, you have to use chemical, and the element, the bonding of element will be broken down. So you cannot get information. It means a lot of information can be lost. But if you use X-ray absorption spectroscopy, you can see the speciation of the element in your sample, what kind of compound of this element, how it binds to the other element nearby. For X-ray absorption spectroscopy, we excite or we shine the X-ray, incident X-ray to the sample, and we get the signal. This is the spectrum. The spectrum, we can get the first information is called X-ray absorption near edge structure. This is can give you the information of oxidation state of your sample. And for delay, the peak here, you can get the information of the element nearby. Like for this one, suppose molybdenum is in the core, and what is the bond length of the molybdenum to the oxygen neighboring atom, and how many oxygen that bind to your sample like this. So it's very useful technique. And this is, you can combine X-ray fluorescence with the X-ray absorption. You can get result of element content and oxidation state of your sample at the same time. Like for this one, you can, if you use the micro X-ray absorption, you can choose which area of your sample, like in the leaf, where is the area that you want to get the information, or you can do mapping to get the distribution of the sample. As I mentioned you that the oxidation state of sample can be distinguished by X-ray absorption spectroscopy. Like for this one, if you would like to probe manganese, this is manganese 3 phosphate. Here is the spectrum. The spectrum is kind of shift into the bright energy, but for manganese 2, oxide will be the energy is lower to excite. So if your sample have different oxidation state, the spectrum will be totally different, and also the feature, the shape of the spectrum is also different. So you can use this shape at the tail of the peak of the spectrum to measure the exarch as well, to see the bond range and the neighboring atom. This one is the example of XRF measuring elemental distribution at the endosperm of 2 days germinated rice. You can see that the rice spout is coming, so we can see the localization or co-localization of each element in each part of the endosperm, like iron, the red one. The iron distribution is in here, but manganese is also quite close to the position of iron, and the copper is in here, totally different as well. Also, if you do the analysis of the toxic chemical like arsenic, you want to distinguish how toxicity of the arsenic in the rice grain. Like for this one, arsenic 3 and arsenic 5, you can measure not only the arsenic form, but it can give you the information of the arsenic in the the organic form or inorganic form as well. So the toxicity of the organic form is less than the inorganic form. So for this one, the hotspot 1, 2, and 3 contain different form of the arsenic, and you can measure further using the spectrum analysis to see the percentage, to compare the percentage or the content of arsenic 3 and arsenic 5 in each position as well. The next beamline is beamline 4.1 in infrared spectroscopy and imaging. This one is combined technique with the infrared spectrometer with the microscope. So you can get the information of the size of the sample using microscope, and you can select which area that you want to get the chemical to identify the chemical mapping of the sample, because this one can measure the organic substance in the sample. You can get the information of the functional group like CO2, CHO, NH, C double bond, all like that. And if you use the synchrotron, you can get a very nice shape of spectrum if you deep down into the area of 5.5 micrometers square. But if you use the conventional FTIR microscope, spectrum will be very noisy. So this technique, you apply infrared into sample, and if the functional group is the specific functional group absorb the energy, you can see the spectrum of energy that's been absorbed. So the peak that we get from the spectrum will tell you the information of the functional group like OH, CH at around 300 wave number, C double ball N, C double ball C, C double ball O at different wave number. So this is like the fingerprint of the functional group. And for the biological molecule is such a complex molecule. It's not like one compound measuring. So we can use FTIR to be the fingerprint pattern of the sample. Like for protein, we will see the spectrum around here at the CH group, and also the big peak at A-1 and A-2 of NH bending. And for lipid, we can see the lipid peak at CH2, CH3 stretching and CC stretching in the spectrum. And for nucleic acid, we can see P double ball O stretching. Also, for carbohydrate, we can see the big shape of OH stretching and CO stretching. So if we do mapping or scan at the position in, we prepare sample by cross section first to like a 4 micron thick of sample, and measuring FTIR, measuring under microscope. Automatically, we can get the full picture of biological molecule localization. Like for this one, the carbohydrate, we back to carbohydrate here, we can do the integration of the peak of CO stretching here. When we integrate peak, we can see which area have higher intensity of this CO peak. So we can see that the carbohydrate is localized at the vascular bundle, and also lipid CH, CH2, CH3 group and C double bond O group all together. We can see the localization of lipids at the vascular bundle as well, but not for protein. Is very close to the epidermis. And if you have like a polymer BRIN, for this one is a starch and polyester BRIN, we can see how we BRIN is good bending or poor bending by using the ratio of OH and C double bond O. So if the good bending, the color of the analysis will be like this. And for the section, the cross section of the thin film, like the food packaging film, there are a lot of layer in the film. So we can measuring one by one of the film and to see how different of the polymer composition. For this one, if you get spectrum like this, we can use the database to do the analysis to specify what kind of polymer in this peak. So this is easier than a complex biological sample, because this one is a polymer is only one composition. And also for the medical application, this is my work. It's about differentiate embryonic stem cell into neuron stem cell. We tracking how changing the biological molecule from embryonic cells into neuron cell. We can see that the protein is totally different. The alpha helic structure of the protein increase significantly when it's changed to be neuron cells. And we can use the program analysis called PCA, or principal component analysis, to distinguish between group of sample as well. So this program we provide user also, and we have the other program is called cluster analysis to see how similar the similarity between a sample. If you have more sample than this one, like you have 40 or 50 or sample, and which sample is close to each other and which one is very far from the other. So you can use this cluster analysis of FTIR spectrum. And this one is the cancer diagnostic. So we section the cancer cells and to see where is the cancer cell deposit. Why not need to be use any chemical, use only FTIR spectrum to distinguish between area of the cancer cells. For the last beam light is the deep X-ray lithography. This one we can use it to make a micro part. You can see it's a very small gear and very clear shape, and you can do a 3D shape as well. For this one, one of application is to do the to make a moisture, very sensitive moisture sensor. So you can see here is a 10 micron. And this one is the temperature sensor. So it can be used for the lab on the shape, very small, like a thumb size. This one is the metal oxide gas sensor to use on a plastic foil platform. So it can be used for hydrogen sulfide, carbon dioxide, and ammonium sensor as well. And also now they do the PCR lab on the chip to use it to amplify the DNA, very quick and very small one. So if you want to use the Synchrotron beam light, you can go to this link and submit your application form, and will be the user of it will be contact you after that. Okay, this is all of my presentation. Questions are welcome.
Okay. Thank you very much, Dr. Waraporn, for your presentation. Quite nice presentation. Is quite what kind of application that Synchrotron can be used for the life science. So many kind of imaging that can be used for maybe for us for publication for Q1. I think it's already much more than enough. So maybe we can go to, actually you still have another 15 minutes, but I think it's fine. Then we can go to the next keynote speaker. Dr. Asitus, can you show our the third keynote speaker introduction? Personal data. Okay. Okay. Thank you very much. There will be our last, but not least, third keynote speaker. She is Dr. Ratih Asmananingrum. She is the head of research center for the genetic engineering of BRIN Indonesia. She is graduate from Bandung Institute of Technology, and her expertise will be protein modification, biosimilar, antiviral, and anti-cancer agents. And her last recent publication will be called Nanoparticles in UNP-based apta sensor for enteropathogenics associated coli detection. And the second one is the in silico design and validation and CRSPR Cas13A systems as a potential for in Indonesia. And the third one, codon optimized human papillomavirus types 52L1 virus like particle in Pichia pastoris, BG10 expression system. And the last one is the dynamic circulating SARS-CoV lineage in Bogor and surrounding area reflect a variant shifting during the first and the second waves of COVID-19 in Indonesia. So Dr. Ratih Asmananingrum have 30 minutes to do presentations. The floor is yours.
Okay, thank you. So, do you see my slide?
Yeah, we can see your slide.
Okay. So, good morning, Dr. Muhammad Rifai as head of Research Center for Accelerator Technology, distinguished speakers, Dr. Somchai and also Dr. Warapon, distinguished panelists, ladies and gentlemen. Warm greeting from Cibinong, Bogor. First of all, I would like to thank you for the opportunity that has been given to introduce research organization for life sciences and environment. And the topic of research activity that related to the application of the use of Synchrotron. On behalf of Research Organization for Life Science and Environment, I apologize that our chairman cannot attend this event because there are other agendas that cannot be postponed. Ladies and gentlemen, we are currently facing various kinds of global issues that require the collaboration of various parties to find solutions. We are in the era of pandemic, which although according to WHO, we will be approaching the end of the pandemic. But there are still many problems related to COVID-19 that need to be overcome. We are also in a situation of climate change, food shortage, and environmental solution. All of these issues require a cross cooperation from all parties in each country to work together. Climate change has had an effect on two of the most important aspects of human life, food and health. We are faced food shortage due to crop failure, triggered by drought, plant pests and disease, and decreased arability. In health, infection from factors are also increasing due to global warming. The problem of clean water also causes waterborne disease outbreak. And all of the issue, actually one of the effort to overcome this problem is through a sustainable use of biodiversity. Indonesia is a mega biodiversity country with diverse animal, plant, and microorganisms. Biodiversity is essential for the process that support all life on Earth, including human. I'm sorry. Without a wide range of animal, plant, and microorganism, we cannot have the healthy ecosystem that we rely on. Healthy ecosystem, of course, will be more resilient to climate change and so more able to maintain the supply of ecosystem services on which our prosperity and well-being depend. So as a research institution, BRIN strives to play an active role in management of Indonesia's biodiversity. So BRIN established research organization for life sciences and environment. So this is the organization chart of National Research and Innovation Agency of Republic of Indonesia, which consists of 12 research organizations, including Research Organization for Nuclear Energy as our organizer
of this webinar. Voluntary Research Organization for Life Sciences and Environment has 3 goals. The first is how to answer the global challenge by mastering the key technology to be able to contribute to problem solving. The second is how to support sustainable development goals. And the third is how to transform national industrial development from mineral mining to data mining. There are 2 key strategies here, partnership and also connecting island, including field station, capacity building, and community development. To achieve the goals, we perform biodiversity-based activity from discovery, collection, conservation, and rehabilitation. The genomic and protein data from animal, plant, and microorganisms are used as basis information for sustainable use. By 2023, BRIN plans to build 25 field stations in potential locations in Indonesia, with 12 existing field stations and 13 new field stations. The objective of field station establishment are to improve management of biodiversity, to strengthen economic development planning, and to improve the quality of the environment. There are 4 goals to be achieved: bioprospecting, biodiversity assessment, biobank, and community empowerment. To facilitate the activity and goals in biodiversity, Research Organization for Life Sciences and Environment divided into 8 research centers with 4 research scopes from inventory, conservation, sustainable use, and environment. There are 2 research centers with basic science and 6 research centers with applied sciences. The basic science are Research Center for Biosystematic and Evolution and Research Center for Ecology and Ethnobiology. The applied science are Research Center for Plant Conservation, Botanic Garden, and Forestry, Research Center for Environment and Clean Technology, Research Center for Genetic Engineering, Research Center for Applied Microbiology, Research Center for Applied Zoology, Research Center for Biomass and Bioproduct. I will give you a short description about each of the research center. So the first research center is Research Center for Ecology and Ethnobiology. The research scope is to monitor and mitigation the climate change effect, carbon capture and sequestration, and also the reduction of carbon emission, land and ecosystem rehabilitation, strengthening management of invasive alien species, and also acquisition and implementation of traditional knowledge for inclusive economy. There are 20 research groups here, and the head of Research Center for Ecology and Ethnobiology is Dr. Anang Ahmad. The second research center is Research Center for Environmental and Clean Technology. The scope of research is environmental management and assessment, pollution control, pollution prevention and reuse, and also environmental quality recovery. The head of Research Center of Environmental and Clean Technology is Dr. Sasa Sofyan. And the third research center is Research Center for Biosystematic and Evolution. This scope of research of this research center is from inventory. So this is a very fundamental stage, analysis and assessment, and also synthesis process. The head of this research center is Dr. Bayu Aji. The fourth research center is Research Center for Plant Conservation, Botanic Garden, and Forestry. So the scope is many, from genetic conservation, tree health, domestication, seed conservation, ecopsychology, and many. And there are 27 research group. This is the very big research center, and the head of research center is Dr. Andes. The research center for biomass and bioproduct. This is more, the scope of research is more biomaterial actually, and the research center focus on three main factors: bio-based, bioactive, and biocompatible.
I'm sorry, Dr. Ratih, your slide doesn't move.
Oh, I'm sorry.
The environmental and clean technology right now. It's still the Dr. Sasa.
Oh, okay.
Okay.
I will end. Sorry. I will reshare my presentation. Okay, sorry. Do you see my slide, Pak Rifai? Yeah, okay. So the research scope is like utilization of biomass. And the head of the research center is Dr. Ahmad Hanif Dawam. And next is Research Center for Applied Zoology, with research scope is from experimental zoology, animal health and disease, applied entomology, and animal biotechnology. And the head of this research center is Dr. Evi. And then the Research Center for Applied Microbiology. The head of the research center is Dr. Ahmad Fatoni. The research scope is enzyme actually, and also microbiome, metabolic compound, microorganism utilization, bioconversion, and also bioremediation. And the last research center is my research center actually. Research Center for Genetic Engineering. As the title of the research center, we are focusing in genetic engineering. So from gene discovery to gene delivery, molecular detection, metabolic engineering, protein engineering, and host engineering. We have 11 research groups, and we divide it into two clusters. Health cluster and agriculture cluster. So we are work with many plants and also many virus, bacteria, and mammalian cell, and also the DNA and protein engineering. So what kind of program in research organization for life sciences? We refocus the research program. We established a program for disclosure and utilization of Indonesian biodiversity that divided into 5 work breakdown structure. The first is to disclosure of biodiversity from animal, plant, and microorganism. The second is whole genome sequencing and partial DNA and protein sequence. And the third is utilization of biodiversity. So this is more like bioprospecting actually. And the fourth is environmental impact on ecopsychology and population diversity. And the fifth is rehabilitation for endangered species population. So this program output is 50 new species. And the second big program in research center, in research organization for life science and environment. I'm sorry, this slide is in Bahasa, but I will try to translate to our distinguished speaker. So this is the program for plant conservation, endangered plant conservation. This program divided into 3 WBS from exploration and conservation, ecology and restoration, and experimental botany. And the output from this program is 50 endangered species. So I will give information about the research center for genetic engineering that's many of our research can also use the synchrotron actually. So as a research center in sustainable use scope, we focus on genome and protein for bioproduct application. We perform gene discovery, gene delivery, molecular breeding, protein engineering, host engineering, metabolic engineering, and also molecular detection. So there are some research topics in 2022 in green biotechnology and red biotechnology, as I mentioned before, because we divide the research group into 2 clusters, agriculture and health. So we perform the research for green biotechnology and red biotechnology. How is the application of synchrotron for virus research in research center for genetic engineering? Actually, I just learned a lot from Dr. Warapon actually in previous presentation about the application of the X-ray for the life science research. In green biotechnology, we are working with many plants actually from ganyong, coffee, banana, rice, also chili, palm, and also the sugar cane, cassava, corn, and many more. And for in red biotechnology, actually we are working with genetic engineering to construct the vaccine with the subunit vaccine like the Omicron spike COVID-19 vaccine, and we also developed the pseudovirus and pneumococcal vaccine, human papilloma vaccine virus-like particle, and also the monoclonal antibody. We can see the more detailed information here. So I also apologize that in this slide is in Bahasa. In research center for genetic engineering, we developed the pseudovirus research. So we synthesize a non-virulent virus that can bind into the spike receptor. The internalization pseudovirus by human H2 expressing cells was performed using a fluorescence microscope. But the structure of pseudovirus has not been further characterized, including how it binds to the receptor. X-ray diffraction analysis or maybe imaging is very necessary to determine the process of the protein-protein interaction. This pseudovirus platform has been used to determine the anti-SARS-CoV-2 from Indonesian plant extract. So we don't need the biosafety laboratory level 3 to perform this assay. We also perform the SARS-CoV-2 live culture actually to challenge the anti-SARS-CoV-2 agent or antibody from the serum. We isolate the virus from Indonesian isolate from clinical samples. The virus already sequenced, but the variant structure and the spike protein have not been characterized and analyzed. So maybe this is also the chance to study more about the variant structure using X-ray diffraction. And also we developed the virus-like particle for human papillomavirus. Human papillomavirus is the virus that causes more than 90% of cervical cancer. We are developing virus-like particle that can be used as a vaccine in the future. The form of the virus-like particle need to be compared actually to the native variant, because native variant is composed with two major protein, L1 and also L2, but the virus-like particle only compose L1 protein. So actually we have to compare the characteristic of this two particle and study about how the particle can induce the immune system. In SARS-CoV-2, we developed the subunit protein for vaccine. We designed a trimer protein that consists of spike SARS-CoV-2 that fused with foldon to mimic the native spike binding into S2 receptor. The three-dimensional structure determination is of course very important to characterize the trimer formation, because this is the key of the immune system response. And also this is the pneumococcal conjugate vaccine. This vaccine is very unique because the protein is conjugate with glycan. So the structure actually is very complex. So we cannot just characterize by using two-dimensional electrophoresis, for example, because we need further protein characterization to check the successful of gene cluster construction, because there are 8 genes that responsible to capsule formation here. From previous presentation, maybe we can determine the carbohydrate content using infrared imaging, or determine the bilayer seed using small angle X-ray scattering. And the next research is for biosimilar insulin. Actually, this is a very old protein. Yeah, we know that insulin is used for the diabetes therapy. One problem in insulin, we have to convert the proinsulin into the mature insulin, and so far we already produce the proinsulin, and we try to convert the proinsulin into insulin. By using the two-dimensional electrophoresis, we have the correct size. But
Dr. Ratih, another 5 minutes left.
Okay, but we don't know if the structure is correct or not, because the uniqueness of the protein, it is small protein, but consists of 3 peptide chains. So that's why this is very difficult to get the mature insulin. And also the same case with protein core of hepatitis B. We produce the protein in E. coli, but the formation of the protein never been compared before. In the protein is produced in E. coli and Lactococcus lactis. So we have to compare the three-dimensional structure in these two hosts actually. And next, we also developed the aptamer for biosensor application actually. And we have to characterize the folding of the aptamer to detect the hepatitis B and also the enterobacter. So that's the application in a health cluster. How about in agriculture? Currently, we are working with sugarcane. The big problem in sugarcane is the productivity actually, because there are many diseases that causes by viruses, fungi and bacteria. So we are currently looking for the mosaic virus, sugarcane mosaic virus, and also sugarcane streak mosaic virus that can decrease the productivity in sugarcane. We can compare the two viruses actually and study the virulence factor to sugarcane. And also the same case in banana. Banana also has problem with banana bunch top virus that can decrease the productivity of the banana. There are many BBTV type actually, and we have to study more about the structure of the variant and the effect to banana. And also the fungi, the fusarium that decrease the productivity also. There are many types of fusarium, so we can compare the most dominant fusarium in Indonesia actually. And also like the previous presentation before, that we can also study the component in the rice actually. Actually, we developed the iron and zinc high rice for stunting in Indonesia. So we can study the composition of rice by using the Synchrotron. So if we talk about the biodiversity research, it cannot be separate with facility actually in infrastructure. BRIN is fully committed to try to meet all the need of research infrastructure. We built various facilities such as the International Standardized National Depository. So we have many collections from plant, microorganism, and animal here. And also we develop the genomic building for the genome and protein research. And also we develop the BSL-3 and animal BSL-3, and also the cGMP facility to support the recombinant protein activity. So to conclude, many research topics in research organization for life sciences, especially in Research Center for Genetic Engineering, that can use synchrotron application. Further collaboration is needed to perform the analysis. So thank you very much. And that's all my presentation.
Thank you very much, Dr. Ratih, for your nice presentation. It's quite interesting about how the structure of the research organization of the nature and environment. So also the research and development regarding about how to use the Synchrotron application. We still have three panelists that will be have a discussion. So I would like to invite three panelists. First, Dr. Taufik, Dr. Vina, and Dr. Satyanugraha. So first, I would like to I'm sorry, wait a minute. I would like to invite Dr. Taufik Purnanugraha to have a discussion regarding about the Accelerator Synchrotron and also the technology and applications. You have 20 minutes to have a raised question and then have a discussion regarding about which research and development that can be applied for the discussion under your research center, something like that.
Thank you very much, Dr. Rifai. My name is Taufik. I'm from the Center from Applied Zoology. I'm very interesting about the Synchrotron technology. I mean, thank you very much for the amazing presentation by Dr. Sonchai and Dr. Warapon. From the presentation, we could see the Accelerator Synchrotron is like a Swiss Army knife of multiple X-ray and multiple spectral diagnostic research applications. I know it's kind of discussion, but in our field in applied zoology, one thing that really interesting for my research group is about for the bioimaging aspect of the Synchrotron technology. The existence of Synchrotron with various bioimaging applications can accelerate research in Indonesia. One of our current interests is for the biomimetic, for the microstructure of many species of fauna in Indonesia. I have some questions regarding the application of tomography. What is the limitations of the applications of Synchrotron tomography compared with the conventional device like the microcity? And as I see from the slide, is the facilities can be customized. For example, the beam size and the platform size. From the slide, I see this limitations with the sample size. You have to have like small sample. While in our research, you want to do like non-invasive reconstruction from bigger samples, like from the whole skull or something. Is that also possible using the Synchrotron, or we could like have to use like another device like the conventional microcity? And also for the spectroscopy applications, what is the key differences between the tools with Synchrotron with other tools like Raman microscopy? I see like many similarities with the thing. And the last that I want to ask is about, do you also have experience, or do you also conducting for the fast contrast X-ray imaging using your Synchrotron? Thank you very much.
Thank you very much for the question. Maybe Dr. Somchai or Dr. Warapon can answer the question.
Okay, thank you very much. Okay, for the question, I don't know the detail, but what I can say is, for the bigger, the beam size for tomography is 10 millimeters by 4 millimeters, so it's quite small. It's better for small sample. We can do a bit bigger sample by collecting each image and then combine them, use the data to combine them. But for large sample like animal, there are some beam lines in the world, like in Australia. There one beam line there. They have very long beam line, because the beam size original from the electron beam is very small, so they have very long beam line to make the beam divert into very big beam, so they can use to try the X-ray to big sample like animal. They can do tomography. I can find information for you. Yeah, you can try to contact them. Yeah, and also in Canada, they also use Synchrotron to do the tomography. If I remember correctly, I think it's that there are only 2 beam lines in the world. Probably might be in Europe somewhere.
Okay.
But as far as I know.
And how about the acquisition time?
Acquisition time
can be faster than the microcity. regarding the acquisition?
Yes, really. I'm not sure. It's less than. I think it's less than one millisecond. No, one. Let me check.
To generate the detailed scan of like specimens, how long it will takes. Like you say in the chat, it's like an up to 1.5 1 micron resolutions.
I don't know the number, but I think it's as I remember correctly. I did my experiment. It takes I think 1 millisecond per image.
Oh, per scan. Oh, so
per image.
Yeah, per image. Yeah, because you capture like multiple 2-dimension image and then you stack it to like 3 dimensions. So like for 900 stack is really fast compared with.
Yes, I'm. Let me check. I find that number and then I put it on the chat box.
Because like this is technology is something that's very new for me. I mean, like we always think about particle of like accelerator is something like really sophisticated, like for physics research and something, and we just like learn today like there's also a lot of applications for other things, including like for photography and etc.
But actually, Synchrotron is not that. What should I say? It's not that really advanced. It's just a source. It's such an X-ray source, but it's really intense and really and the energy is tunable. So it's a perfect source for experiment.
You mentioned about 1.3 gigavolt about the energy is GAV, sorry, 1.3 GAV. How for the tomography application is the final energy is also that big? I mean, like for microcity we usually use like 160 kilovolt, like for the X-ray. Is it the same or it's
okay? It's different. Like I mentioned, because the synchrotron source is low energy, it's 1.2 TeV, and for tomography beamline, they use the energy from 5 keV to 20 keV. So it's used for not very thick sample.
Okay, so it cannot be applied for very thick samples. Okay, thank you. I mean, for microcity, I think what we order is something like 160 keV. And regarding the another questions about the difference between like the Raman spectroscopy with the synchrotron applications.
Okay, I let Dr. Varapon answer.
You mean IR microscope and Raman microscope, right?
Yeah.
Actually, it's the complementary technique. For IR, you can see the absorption of the functional group that have the dipole moment, but forอะไรนะเขาทำอะไรนะ IR กับอะไรนะ Raman. Okay, for Raman, so the functional group is not need to be like dipole moment. So the inorganic bonding, you can get the signal from inorganic bonding as well. So for the IIs, mostly you can see the functional group of the biological molecule like CH, CO, C-double-O, like that. But Raman, for the inorganic, you can get a lot of information like for carbon like this, and the diamond, you can get information of diamond.
Thank you very much. And I still have like a lot of question actually about the resting. You see like from the slide you have like 2 months of resting for the facilities. Is that just like because it's happening to be for the summer break, or you choose that particular time for resting the facilities? I think June to August, right?
Okay. Actually, it depends on our. It's a maintenance. We usually shut down the machine for 2 or 3 months to do the maintenance, and also sometimes we have to install some equipment, and it needs to be shut down before the installation. And also, because this year is we have the energy crisis. We have problem with the budget. So because the electric bill is very, very high.
I mean, how just a rhetorical question? How expensive is like for the facilities? I mean, for the Synchrotron facility.
Okay, the operation costs mainly come from the electricity, which is about, I don't know, in Thai baht, it's about 3 million baht a month.
I have to Google it. Oh, yeah, it's quite big. And how about for the building, the facilities itself? It's like really, you said like it's actually not like an advanced technology. Is that very expensive? I mean, compare like we just bought, I think the cryo EM is like really one of the most expensive things that we have today in BRIN. But compared to that, how expensive is for the Synchrotron facilities?
Okay. I got the cost estimation 2 years ago for the new machine for 3GV machine. It's about $1 million $1 million for 1 meter.
Oh.
Then, for example, our Synchrotron machine is about 300 meters. So the cost of the machine, including not the building, only the machine with the beam line, is about $300 million.
Oh, that's that's a lot. Yeah, but thank you very much for the presentations and introduce us to this technology. Even is kind of old, but for us it's still something new. And hopefully we could like have like future collaborations and even like visiting to your facilities to see more detailed applications of the Synchrotron. Thank you very much.
You're welcome.
Mr. Rifai.
Thank you very much, Dr. Taufik, for your nice discussion. We have another two keynote speaker. Two panelists. Maybe the next one will be Dr. Vinamrita Laxmi from the Research Center for Applied Microbiology. So, Dr. Vina, you can start the discussion.
Okay, thank you very much. Good afternoon, everyone, ladies and gentlemen. Actually, today is a very interesting presentation about synchrotrons, and from our Research Center for Applied Microbiology, one of our main research is about enzyme. So I think it's very closely related with the protein structure. If we want to know the 3D structure of the enzyme by using X-ray usually. Protein crystallography. So maybe for the background of my research itself is I'm very interested in the protein engineering. So I have performed research about the mutation of enzyme, and then we need to analyze the role of the amino acid for the improvement of its biochemical characteristics. And then it's very useful if we know the protein structure of our enzyme, but unfortunately, not many 3D protein structure is already done or reported. So I hope by this discussion today we can have a future collaboration related to that. And maybe my first question is about you. Previously, Dr. Somchai already presented about facilities related to Synchrotron before, and many instruments related with that. So if we want to research about the 3D structure using protein crystallography, do we need so many instruments if in the future we also want to build that kind of facilities in Indonesia? Maybe this is one question. And for second question is, for protein crystallography, we need to crystalline the protein before we get the 3D structure. So what kind of purity that we need to crystallize our sample or our protein? Do we need a very pure, or can we use only crude extract of our protein? And then for crystallography of the enzyme itself, it is needed a certain instrument to crystallize the protein. Maybe that's for me. Thank you very much.
Okay, let me answer the first question, and then Dr. Warapon answer the second
briefly
one.
explain.
Okay, for protein crystallography, yes, you need your protein to be crystallized before conducting the experiment, and in our facility, I think for crystallization is not very advanced. I mean, for the equipment, but what we have here is we have the robot to help user to crystallize the protein, because as I know, I'm not in the field of the protein crystallography. So as far as I know, the protein is normally, when you purify the protein, you get very small amount of protein, and when you do the crystallization, you usually vary the concentration of the solution, and you need a lot of you need different concentration to crystallize your protein. You need a well of 96 wells and vary concentration, and then you expect some of them will crystallize. You will get some protein crystal from some wells, and the robot comes in to help you minimize the amount of protein you have. So you, when you use the protein, you can use the robot to drop like a tiny drop of protein, like microliter protein, into each well. So you have more chances to get your protein crystallized. So the robot helps a lot. Yes, you might need it if your protein is very difficult to purify. The robot might help.
I see.
And for the second,
okay, I see. How about
question? Yes.
Please. What to answer? What is the question that you want to ask?
Also, first, there is so many instrumentation that before explained by Dr. Somche. About the protein crystallography itself, do we need all of that instrumentation or not all, maybe just some specific instrumentation, something like that? For my first question, Dr. Somche.
You need the specific refrigerator, like a 20 degrees Celsius refrigerator to cool down the protein to let it be crystal very slowly. And the microscope, the stereo microscope, to see what the protein been crystallized already or not, and any instrument. Actually, it's not need a lot of instrument. Because if you do not have any instrument, you can drop it by your hand 1 millimeter, well, 1 microliter of your protein, and the chemical you will add it by yourself, and it will take time because it's 96 well in one plate. But if you have robot, it can do for you very quick and very precise. And if you do not have the refrigerator, you can keep it in room temperature, or you under the air conditioning temperature as well. But the crystallization may be occur very slowly, then leave it in refrigerator.
I think my colleagues usually use 4 degrees Celsius or 18 degrees Celsius.
It depends on protein, how big of your protein is, and you can find the suitable temperature in the literature review.
Okay, so for make a protein crystal, we don't need any special instrument. But after we got the crystalline of our protein, and then later we can use the X-ray, something like that, Dr. Warapon?
Yes, that's right. But before you get the purified protein, you need a lot of instrument to pure your protein first, because it needs to be pure. Unless it will be not very easy to do the analysis. And you mentioned that you will buy the cryo-EM. So you get the very nice structure from cryo-EM with not need to do the crystallization anyway. It might be better.
Yes,
But you get the whole shape of it, but not the molecular atomic, the level of atomic inside is not get that information. You can get that information from Synchrotron, but you can see the overall of the shape of your protein with cryo-EM, and it's faster.
Yes, that's interesting, because from what I've read, is it also better if we have a combination between X-ray crystallography and the cryo-EM, I guess. In your opinion, is it correct, or a combination of the cryo-EM and the X-ray crystallography for the analysis of 3D structure of the protein? Or we don't need that? I mean, just cryo-EM itself, or only X-ray crystallography is enough?
I think you will get a lot of information of both technique. And for the detail of the active size with the substrate and your protein active size binding, how packed, how well of it binding, you will get more information from protein crystallography.
I think as I know that I've been to the diamond, the synchrotron in the UK. They have several machines of cryo-EM, but they still have the beamline is still necessary to do the protein crystallography, and they use both technique to study the protein structure. And also to answer your question, if you would like to collaborate with us to do the protein crystallography, maybe it's better to do the crystallization here, because I saw my colleagues when they use the synchrotron in Taiwan, they crystallize the protein here, and then they have to put it in the cryostat and send it to Taiwan. So you need a cryostat to keep your protein crystal, because it needs to cool down all the time during the transportation. So imagine if you have your protein, you need to carry the cryostat. It's like 15 kilogram with you on the plane, and then come to Thailand. So maybe it's better to carry your purified protein and do the protein crystallization here at SLRI and collect the data using our beamline.
Okay, noted, Dr. Somchai. So if we have a future research collaboration, I will certainly do that. Thank you very much, Dr. Somchai and Dr. Waraporn.
Thank you very much, Dr. Pin, for your nice discussion. So it's been the cryo-EM and the synchrotron accelerator we can. So thank you very much. Oh, I'm sorry, still mute. Thank you, Dr. Fina, for your nice discussion. So I think the cryo-EM and the synchrotron accelerator will be complementary each other between the data analysis within the structure and the micrograph from cryo-EM, and then also the atomic structure from the synchrotron accelerator. I think it's quite nice research cooperation in the future regarding about the protein and data analysis in the future. Okay, I think still have 5 minutes, but it's okay. We can go to the next panelist. Will be Dr. Satya Nugroho from Research Center for Genetic Engineering. You have 20 minutes for discussion and then raise question to the keynote speaker. The floor is yours.
Thank you very much, Dr. Rifai. Yeah, thank you very much for Dr. Somchai, Dr. Waraporn, and also Dr. Ratih, yeah, for the presentation. I think today's presentation give us a lot of insight, yeah, on the application of cyclotron. Before this event, actually my knowledge is very limited. So I just understand that there is a X-ray crystallography, and it was Franklin for determining the DNA structure. So, but by listening to your today, your presentation, we understand that the application is quite wide. Yeah, so much wide array of application that can be done in terms of life sciences research. Yeah, it's so amazing. And this is only using 11 beamline, and you are planning to improve the beamline into I don't know how many beamline by using a new you are establishing a new synchrotron with a bigger capacity, like 3-point-something GeV in comparison to today's, which is 1.2 or something like that. So as explained in more detail by Dr. Waraporn, the application is very wide, yeah, in life sciences research. As Dr. Ratih always, there are a lot of research going on in her research center. I actually, I'm part of her research center employee. So I work for Dr. Ratih. And my focus is in plants. Yeah, plants. And well, there are a lot of aspects in plants that we are currently investigating, which could be help supported or accelerated the research result by applying this technology, your facility. Okay, so among others are the attempt to, change cell wall composition. For instance, cell wall composition consists of lignin, cellulose, and so forth. And there is one research that attempted to change the cell wall composition for different purposes, for bioethanol, for instance, for particle board, which will change the composition of the cell wall. Other research that in my mind is deterioration of food crops, of food products. Dr. Warapon also mentioned about that. Yeah, I think that's very interesting, because food loss is very important. Yeah, we cannot afford food loss in time of difficulty like this. So studying the food loss, food deterioration, I think we can try to manage to improve the ability of food products to stay fresh. Yeah, for instance, yeah, something like that. And also, there is also research on the application of biofortification, Dr. Ratih mentioned. And we are currently focusing on iron and zinc, for instance. We use an equipment which is called XRF. I don't remember the brand name, but it is XRF, and we measure the content of iron and zinc in rice. So for the purpose of biofortification. And I think there are many other research that can be done using your facility. Yeah. So the question is, because you already mentioned, Dr. Somchai, regarding traveling time, because our samples are mostly biological samples, which can be considered as alive. Yeah, it easily destroyed. So maybe this is also very timely for Dr. Rifai to think about such a facility in Indonesia, because traveling time will be bad for our samples. Yeah, traveling, sample handling, and so on. Yeah, so maybe Dr. Rifai can start establishing similar facility in Serpong for this purpose. So there are questions. Yeah, I would like to mention about collaboration, which is free. Yeah, this is also very nice scheme that you offer. Yeah, how many countries have already response offer for collaboration using the free facility, and what that they usually have for their experiment? Yeah, and in terms of the equipment, because such an integrated equipment which can measure several analyze several for several purposes, whether for each analysis you need a different sample, or the samples that is being used for one analysis degraded after one analysis. In that case, maybe you need to provide several samples the experiment. Yeah, I think that's all. Maybe you can answer first, and then I will come up with other question. Thank you.
Okay, that's quite a few questions. Let me answer. Okay. Okay, your last question about other countries. We have quite a few actually. We have some researcher from Malaysia, from Singapore, and many users from Indo actually, from Indonesia. They're from Surabaya. I don't remember the name. And also we have some user from Malang. Yep. And we also have users from Brunei, from Philippines. There are quite a lot of users from ASEAN. And also we collaborate with other users from any part of the world. Like we have users from Germany, from Australia. And for the new machine, we're going to have I think 20 something beam lines. So it's going to be more techniques available for more users. Because in our current machine, we have 11 beam lines. It's quite limit already because of the area is not that big, and so we can have a few more beam lines, but that's it. We have a lot of requirements from users, especially in domestic users. And let me tell you something about the advantage of the Synchrotron facility is the in situ experiment. Because usually when you buy an equipment like XIF, XRD, you can do some in situ experiment, but you have to buy the equipment from the company. You cannot modify out yourself. For the beamline, for the Synchrotron, you are freely modify your equipment. You would like to apply some voltage or some temperature. You would like to do some stretching to study the structure of polymer, something like that. You freely to do that. So it's really convenient to develop your own chamber or your own equipment. That's the good thing for Synchrotron facility. Okay. And
that's very interesting. So for that case, when you want to change or add different function, you actually establish it in your own workshop. You mentioned that there is some activity for establishing new attachment facilities for different.
Yes, exactly. Yes.
Right. Yeah. So you have your own workshop actually with people. Okay.
Yes. Like for example, in one of our beamline, we develop the heater that we can heat the sample to 1,000 degrees Celsius and do the X-ray absorption to study the structure that change with temperature or something like that.
Yeah. So as I mentioned, I use XRF for zinc and what is the iron contents in rice. Dr. Warapon mentioned about not only zinc and iron, but also copper and other compound. Other
element. Yep.
So yeah, is that the difference between the XRF that we use with your equipment, that your equipment can detect more minerals than XRF? Because I remember that the technician who operated the XRF mentioned that this particular equipment is only established for iron and zinc and only from rice. Because if they use it for different material to detect similar or to detect different minerals, it will be difficult to re-establish. So you really have to reset your equipment and start from the beginning.
Okay. You don't need to reset your equipment. You actually we have some research on studying the contamination of heavy ion like cadmium, arsenic in rice grain. So we control the condition, put some solution that contain arsenic and cadmium, and study where the cadmium or arsenic go into the rice plant, and the advantage of using synchrotron radiation is the lower limit detection. You can go to like a few ppm, so it's lower than conventional source. And as I mentioned, the synchrotron radiation, you can focus the beam into very small beam, so you can study the distribution. You can see where the element where the element is in your sample, and also you can even analyze the oxidation state of that element, not just the iron or copper or zinc. You can study arsenic or any element, especially for transition metal. And you don't need to reset the equipment. But if you would like to measure the quantification, the concentration, you might need to prepare the you need to calibrate the measurement first. You need to prepare the standard that you know the concentration, just like conventional source.
Yeah. Another one for life sample. Can you like a biological tissue, for example, can you do time point data acquisition?
I think what we can do is we have the problem with sample damage as well, radiation damage. Usually what we can do is we can use the cryo stream, the gas, the nitrogen gas from liquid nitrogen. So the temperature is 100 Kelvin. So to cool down the sample. So it helps. It's the same as for the protein crystallography. So they have the problem with the radiation damage as well. So they use the cryo stream to cool down the sample, to prolong the crystal. So we can use the same for the live sample.
So sample actually not that live and have moisture. You have to do the sample preparation first, because in each beamline need sample preparation differently. Like for infrared, if you want to do the mapping of biological molecule, you have to do cross section in 4 micrometer thick very small size of sample, and after you cut it under cryo section, and you have to put it under the KBR window or barium fluoride window, like that is a specific slide that cannot be absorbed by infrared light. But for X-ray fluorescence, if you want to do a mapping or imaging of elemental distribution, you have to cut it thicker, like 100 micrometer thickness of sample, and put it onto the PE polymer, the polyethylene film, that it will not absorb by X-ray light. So both of these sample preparation have to be prepared under cryostat. So the sample will not be destroyed. It will be like intact sample, and you put, after you cut it, you will dry it under ultra high vacuum. So it will attach to the slide very quick. So for the bulk property analysis, like XAS, X-ray absorption, spectroscopy, or SAC-WAC, small angle X-ray scattering, you have to glide your sample to be a powder first, and to make it to be a pellet in a size of 1 centimeter round pellet.
So okay. So the sample preparation is crucial for and has to be prepared accordingly. According to what you want to use for analysis. Okay, thank you very much.
So we have to do a discussion of sample preparation before sending sample.
So how about the Surabaya sample, Malang sample that Dr. Somchai mentioned? What kind of samples are there? Are there biological samples or?
Okay, I don't remember. I think it's material science, not life science. But I think Dr. Wutika is here. He probably knows. Anyway, I find out and then I let you know.
Okay, okay, thank you. I think that's all for now. I think my time is already up. Thank you very much for the enlightenment. Thank you very much for the information.
You're welcome.
Okay, thank you very much, Dr. Satya Nugroho, for your nice discussion. So thank you very much to Dr. Somchai and Dr. Warapon for your explanation. So actually, this the three panelists is quite interesting with some application of the Synchrotron Accelerator itself. So maybe we need some information regarding how can we use your beamlines, what kind of procedure that we need to follow regarding about how to if we need to send the sample, or our researcher can go to the SRE, and how about the accommodation, like transportation, maybe some maybe like research funding to collaboration, something like that. Is there any thing about it? So maybe Dr. Somchai or Dr. Watakron can explain about it. Maybe this kind of scheme, is it possible to have that kind of collaboration?
Okay. Firstly, as I mentioned in my presentation, that our facility is user facility. Basically, you can go to the website and then you apply for the beam time. If you're familiar with the technique or with the equipment, if you don't need any help from our researcher or our staff, you can apply for the beam time, come to our facility, do the experiment, and take your data back home. We don't need any collaboration. If you don't want to, if you need some help, or if you need to collaborate, the first thing I would recommend is send in an email to the beamline manager.
Mm.
If you know that which beamline you want to use. If you don't, send an email to user office.
Mm.
We have the user office. It's the middleman
Mm.
to communicate between user and the beamline manager. And then when you discuss, when the user office will communicate to the beamline manager and find the right person to discuss with you.
Mm.
And to discuss the detail of the samples, and when you finalize and when you make a plan, and then you can discuss whether you would like to come to do your own experiment, or you would like to send a sample and ask our colleague to do an experiment for you and send you the data. That's the way.
Okay. Then I think already clear for our three panelists from the Research Center for Applied Geology, Microbiology, and Genetic Engineering. I think they want to use Synchrotron accelerator in the future for complement their result using non-irradiation equipment with cryo-EM or maybe Raman or FTER, something like that. So I think it's quite a good collaboration in the future to publish some paper regarding about their experiments. So I think our panelists three already finished to discussion. Maybe I'll try to open discussion to the audience. Maybe some of them already tried to put in the chat room. So I think Dr. Somcha already answered it very well. So maybe, there's some, Mr. Rifai raised hand. So maybe you can ask directly.
Thank you, my brother. Okay. Yeah, I want elaborate for my chat info. So I still interested with detector. Yeah. I'm from neutron scattering lab in Indonesia. And in the past time, I joined with the development of neutron diffraction for biological sample in Japan. In that instrument, we use a circular or tube detector, which the sample is put in center of the tube. For
Mr. Rifai, your internet I think is slowed down. Dr. Mr. Rifai, I think your internet is stopped. I think you need to repeat again another your 1 minute to like 30 seconds is lagging, something like that.
Also,
yeah. We cannot hear your voice.
About the detector of X-ray crystallography. In case of neutron crystallography, we use a tube detector, which the sample is put in the center of tube, so the diffraction can be observed. Around the detector. Of course, it's not for fine, like Cyclops in YLL. But in case of X-ray or a synchrotron, cannot imagine what's the shape of detector for crystallography, especially for biological crystallography, because another crystallography maybe look like general XRD. That's it for Mr. Somchai. Thank you very much.
Okay. I'm not sure I can answer your question. But let me give you some information. We do have the X-ray scattering, small X-ray, small angle X-ray scattering, and diffraction, which is, I think, is similar to neutron scattering.
Yeah.
We put the sample into the small tube,
Yeah.
and we hit the sample with the X-ray and use the CCD detector to collect the pattern and analyze the data. And for X-ray tomography, we hit the sample with X-ray beam, the quite big beam, 10 to 4 millimeters, to the sample, because we need to do the imaging, and because the detector we use is a CCD camera. It's like a normal camera, CCD camera, but the CCD camera cannot detect X-ray, so we need a scintillator to convert the X-ray image from the sample into visible light using the I mentioned in the chat box that we use YAG cesium as a scintillator to convert to visible, and then use the CCD camera to capture the image. I'm not sure whether I. Yep.
Yeah. So for both about technique, it use a flat detector. Is it right?
Yes, we use.
Yeah.
Yeah.
Yeah. How about the X-ray crystallography? Do you use also flat detector?
No, we use CCD detector.
CCD detector. Okay, I see. So CCD itself is flat. Is it right?
I'm not sure.
One.
Sorry.
One or two. How many? How many CCD?
How many CCD? For sex, it's one CCD.
Yeah.
It's a large area CCD.
I see. Yeah.
It's, as I mentioned, it's 165 millimeter in diameter.
Yeah. Okay. I see.
Is it enough?
Yeah.
Okay.
Yeah.
Thank you for the question. So, is there anyone want to ask again regarding about the synchrotron accelerator technology and also its application, especially in life science? So, I hope from the younger researcher from BRIN maybe can raise a question. Maybe because yeah, as mentioned before from the panelists, Indonesia want to try to construct synchrotron. Maybe we dreaming something like Thailand has right now, but we still need to prepare our human resource regarding about how to construct some synchrotron facility, and also how the utilization about the synchrotron facility. So in this case, maybe we still not need another 5 or 10 years to prepare some human resources, and then also the technology itself. So I hope maybe some audience want to raise some question.
Let me say something.
Okay.
I can give you the easiest way to collaborate with us. Sending an email to me or Dr. Warapon, and then I find some right person to contact you.
I think it's quite easier to us to contact you and Dr. Warapon regarding about how to use the synchrotron accelerator. Yeah, because the user of the synchrotron quite I think lower right now, right? I think because you need more users to use the synchrotron facility. Yeah. Okay, is there anyone want to raise question? I think usually we have this question to our keynote speaker regarding about the synchrotron accelerator facility and also facility. We try to make a program for the accelerator technology, what kind of accelerator that we need to build or construct in Indonesia, and then how about the resource competence, how many human resource that we have to prepare in the future regarding about accelerator technology development. I would like to know about your experience in your last projects. What kind of accelerator that we need to build in Asia to complement the Synchrotron facility in Thailand? And how about the human resource? How many human resource that we need to prepare regarding about the accelerator technology facility in the future, something like that?
Okay. Yes, okay. I think for the first phase, construction phase, you probably need and physicists, because you need to calculate, to do the simulation of the electron beam. Actually, there are two sides. One is the facilitators side who run the machine. So you need an engineer, you need a physicist to do the calculation, to do the model, and to design all the equipment. And another side is users. What you have to do is you need to collect all the data as much as possible of the researcher in Indonesia, which feel that benefit from synchrotron. For example, in our case, when we consider to build a beamline, what we have to know is what application can be used for this particular technique, and how many research group in Thailand that will benefit from this beamline. This data you need to know first. And I talked to my colleague in Taiwan. They mentioned that when they built their own synchrotron, what they did was they sent group of young researchers, young scientists to abroad, to the US, to the UK, to Japan, and stay there for a year to learn how to design the machine, design the beamline, and learn the techniques. And then a year later, they come back, and now they are in third generation. So they send the second generation to abroad, but the shorter period, because they have the first period to train them, and then the third generation they don't have to send to anywhere because they already became an expert already. Yeah, you can do something like that.
Okay,
but
so
I really recommend that as in my opinion, in my experience, there are many facilities. What they first did was building a beamline in other facility. Like for example, in Taiwan, before they build the new machine, they built 2 beamlines in Japan. And they send researcher to stay there for learning other techniques, learning the techniques, and then come back. And for example, in India, they built a beamline in KEK, in Japan also, and in Poland, they just got the new machine. They also built a beamline in Italy to for capacity building.
Yeah, I think it's good.
That's the shortcut.
Yeah, that's true. So it means we need to prepare some young scientists to go abroad, study there. Yeah. Actually, our researcher now is going older and then going shorter too. So I think we need to kind of find a student, in a colleague, something like that, to learn about the technology, and then send them to study about regarding about construct some accelerator facility, something like that. Okay, and then one more question regarding about how you gather some user information. Do you make some conference or you make some scientific meeting? How
Oh,
can you gather
yes.
some user?
As I mentioned in my presentation, we did a lot of activities like meeting, workshops, training, like
Okay.
more than 10 times a year. I remember when we install a machine, during the installation, we have a trip to universities in Thailand and promote our facility. Even before we have the Synchrotron light, because you need to do that in advance
Okay.
before you have the machine.
Oh yeah, that's true. So it means we need to go to university and institution that will use the Synchrotron facility or Accelerator facility, something like that. And 10 times a year, like 10 times event in a year, is quite busy, I think, for the one institution, something like that. Okay, maybe is there anything else?
Pak Rifai, can I ask question?
Yeah, of course.
Quick question actually to Dr. Warapon. Yeah. You mentioned about the protein characterization, Dr. Warapon. Is it possible to see like the protein-protein interaction, like the protein binding into the receptor? So if we want to see the interaction, do we still need to make the crystallization for broad protein? I'm concerned because sometimes we check the protein interaction directly into the receptor on the cell surface. So if the crystallization for broad protein is a must, so maybe we need to also overexpress the receptor, right? So is it possible to see just the interaction directly from protein to receptor on the cell surface? Thank you.
At the cell surface is not that easy because the protein or the glycoprotein at the cell surface is not being purified. So you will put all the membrane, that is the phospholipid with the glycoprotein and with the protein. So the data of protein crystallography will be very complex and it cannot be characterized at the atomic level. But the other technique that would be interesting to use is a small angle X-ray scattering. So for this one, you not need to do the crystallize your protein. Your protein can be in a solution, and if it folds properly, a 3D structure of it, and when it binds to the substrate, sometimes the structure has changed when it folds with the substrate, right? So the small angle X-ray scattering can see the shape changing of your sample of your protein. Maybe if you're interested in this one, you give me the information of your protein and your substrate, and I will tell the responsible person to talk to you later.
Okay, thank you. I will send you an email for more detailed information about that. Thank you very much.
You're welcome.
Thank you, Dr. Ratih. Maybe another audience want to raise questions, maybe some comments. We still have another 15 minutes. Maybe. If not, I will.
I have.
Okay, Mr. Rifai, want to ask again? Okay.
But this question is actually not a technical question, but about the it look like a procurement question that you said that you send many what I can say, young researcher or young students abroad. How can you guarantee that your young student or young scientist will return back to country? Thank you.
Oh, sorry, can you repeat again? I didn't catch. How can I guarantee the student?
You still mute, Mr. Rifai.
Yeah. How can you guarantee that your young student or young researcher that you send to abroad will return back? Because sometimes abroad, the what I can say, salary it's good, or working atmosphere is better than in our own country. Thank you.
So okay, this one is quite easy. What we do is we sign a contract.
Yeah, that's true.
So they have to come back.
Yeah, the documentation is have a signed contract that they didn't need to come back to home country.
But I think that there's some cases that they got a job and they pay back the money, even it's twice the price, twice the cost. They actually pay the money. But it's never happened in at Synchrotron.
Okay, I think so. Yeah.
And I like to mention that also in Synchrotron community, we have the we call Asia Oceania Synchrotron Forum.
EONSA, you mean?
No,
it's similar to ONSA.
I see. Okay, I see.
It's similar to ONSA, but this one is for Synchrotron. Yeah, and we help each other. The big boss is I think it's Japan, Japan, Korea, China, and Taiwan. And they help a lot. We used to send our colleagues to spend time in Australia for 6 months. My colleague to stay there, to stay at the tomography beamline for 6 months and learn about the techniques.
In case of EONSA, we have no agreement one another institute that we will not what I can say hijack or something like that. How about Synchrotron?
For Synchrotron, we have the membership fee. So we have the budget that we can spend to support support the workshop, because we know that Japan is the first country in Asia that have synchrotron. They're like the master of the synchrotron technology. When they established the synchrotron community, they organize the workshop every year and support our young researcher to stay there for 2 weeks and learn the techniques, like every year. And what happened after they also have the economic crisis, so they asked about the membership fee. So but it's not that much. I don't remember. I think it's about 100,000 US dollars. Oh no, I don't remember. But not much to support our activities.
Okay. Is it now, Mr. Rifai? You still mute? Okay, then you know. I'm sorry about him. Okay. We still have another 10 minutes for open discussion with the audience. If it's enough, then I will return to the MC, Mr. Idrus.
Okay, Pak.
Okay.
Okay, thank you very much to Dr. Rifai and all speaker. Let's give a big applause for our speaker. Ladies and gentlemen, all right, we have come to the end of our meeting. The next agenda is summary and closing. Back to Bapak Muhammad Rifai. Please again, time is yours.
Okay. Before I make a statement, maybe I would like to have Dr. Ratih to make a closing statement from the Life Science Research Organization. Dr. Ratih, the floor is yours.
Okay, thank you. First, I would like to thank to Dr. Somchai and also Dr. Warapon for sharing valuable experience in Synchrotron. Thank you for all panelists also and the committee for organizing this event. Today we learned many things about the facility and application use of Synchrotron in life sciences. The application is very in line with many research topics in research organization for life sciences and environment. There are many analyses that can be applied from protein structure like enzyme, therapeutic protein, biosimilar, and virion structure, composition of the samples, protein, lipid, carbohydrate with no destructive analysis. The challenge is how to maintain the biological sample stability until the analysis. So as the representative of a research organization for life sciences and environment, I'm looking forward for further collaboration in Synchrotron application in life sciences research, especially from Research Center for Accelerator Technology. So thank you very much. And we are looking for further collaboration. Thank you.
Thank you very much, Dr. Ratih, for your nice closing remarks. So thank you very much for your attention for the audience, and then also for the keynote speaker, Dr. Somchai, Dr. Waterkron, and also Dr. Ratih for your nice presentation, and then also our three panelists, Dr. Taufik, Dr. Vina, and Dr. Satya Nugraha for nice discussion. I think for Indonesia, it's still quite a long journey to provide some synchrotron facilities. So I think for the short term and using application, I hope the research organization for nature and environmental can come to Thailand. And then actually you can contact Dr. Somchai or Dr. Waterkron easily. Then they can manage for your usage for the beamlines there. I think I heard from the Dr. Mutikrai last week that there's one beamline is available for us, something like that. I think it's quite interesting for some researcher in BRIN to come to visit Thailand. And I would like to conclude our mini symposium today. This have to be two mini symposium. Last week for the material science and then this week for the life science. But I hope it's not only this mini symposium that can be try to carry out in the future. I hope there's another webinar or another workshop, another conference maybe in the future regarding about the technology. And we still hope we have keep in touch with Dr. Somchai, Dr. Waterkron in the future. So I think we hope we can visit Thailand the beginning of the October with Dr. Edikiri regarding about to see the facility there. Okay, maybe from my side, from Research Center for Technology, we thank you. We have to say thank you so much for your for attendees and then for the keynote speaker, something like that. If Dr. Somchai or Dr. Waterkron want to say something, we can invite you. Yeah.
Okay. I would like to thank you, BIN, for organizing such a nice um, symposium. And thank you for the invitation. I think we've been working with Indonesian researcher for quite some time already, and I hope that the collaboration will be strengthened, and we have more collaboration in the future. Okay, thank you very much.
Thank you very much, Dr. Samjay, for your closing remarks. So I think we can closing our agenda today. So I would like to thank to the committee for organizing this event. So thank you very much for your time and effort. So I would like to conclude, and then thank you very much once again. So Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you. Thank you.
Thank you very much. Thank you very much. Dr. Terima kasih.
Okay, thank you. Ladies and gentlemen, Alhamdulillah, we have a productive and inspiring time together. I hope all of you found the presentation on this mini symposium are informative and helpful. On behalf of all the committee members, I would like to express my utmost gratitude to all of the distinguished participants and speakers for making this symposium run smoothly. Thank you. Assalamualaikum warahmatullahi wabarakatuh, and see you on the next agenda. Bye-bye.